Can you see the screen well? And can you hear my voice well? Okay. So let's start. What are the contents of our lecture? We are going to discuss several topics. Start with the bacteria. Then we will discuss the viruses, followed by the fungi and parasites, including herpes. And finally, we'll just give some hints about the sterilization and disinfection regarding the ophthalmology. So what about the bacteria? Let's start first with the bacteria. So in the bacteria, we need to know what are the bacterial characteristics and the bacterial virulence or pathogenicity, and how do the bacteria do their harmful effect to the body. What are the different stains that used to identify the bacteria and the culture media used to diagnose the different bacteria? What are the common and important commensal ocular flora, because this is commonly asked in the exam? And finally, what are the bacterial classifications and the important bacterial pathogens that invade the ocular tissue? Starting first by some bacterial characteristics. The bacteria is called a prokaryotes. What are the prokaryotes? The prokaryotes are just cells that doesn't have a cell membrane or cytoplasm or ribosomes or what's called organelles. So this is just a simple form of life in which the cell doesn't have any organelles inside it. So although it has a cell membrane, cytoplasm, and ribosomes, but they don't have any other organelles or membrane-bound organelles. So they just contain DNA and RNA. Nuclear DNA not enclosed in a membrane, but lies free in the cytoplasm and which is devoid of organelles. So this is the main difference between the prokaryote and the eukaryote. In the prokaryote, the DNA is not enclosed in a membrane, in contrast to the eukaryotes, and also the prokaryotes doesn't have any organelles in contrast to the eukaryotes. The bacteria reproduce by a form of division called the binary fission. So again, here is the difference between the prokaryotes and eukaryotes. The eukaryotes, like the human cell, like the animal cell, like the plant cell, it has a nucleus. This nucleus has a membrane that bound it, and also it has organelles in the cytoplasm. However, in the prokaryotes, although they have a cell membrane and cytoplasm and ribosomes, but their nucleus is free in the cytoplasm, and they don't have membrane-bound organelles. The cell membrane will be different in gram-positive and the gram-negative bacteria, and this will have some consequences later on. We will mention about the gram-positive and the gram-negative, but since we are talking about the structure, so the structure of the gram-positive bacteria, the cell wall contains what's called the peptide interbridges, and it has some proteins. While the gram-negative bacteria, it has a lipopolysaccharides in their cell wall. This is very important for the pathogenicity. So here, to remember the difference in the cell wall between the gram-positive and the gram-negative, think of one of my presentations which are boring and long. So a long PowerPoint will be your mnemonic guide. Long PowerPoint. So the gram-negative has a lipopolysaccharide outer membrane, so it is a gram-negative. So remember that the lipopolysaccharides is present in the gram-negative. While the gram-positive has a positive peptidoglycan, which is a thick layer. Teichoic acid, it is one of the content of the cell wall. So remember that the gram-positive has peptoglycan and teichoic acid in their cell wall. So let's talk about the basic structure of the bacterial cell. So if this is a photograph showing a bacterial cell and demonstrate its different parts. So start first with the cell wall. Here is the cell wall. The cell wall is a mucocomplex of murmuric acid that allows a entry of molecules of all molecular size, except if there are very large molecules, then it will inhibit the passage of this very large molecular weight molecules. Then it is followed by the cell membrane. The cell membrane is an important osmotic barrier, and this osmotic membrane or this plasma membrane is the site of action of many enzymes, and it controls the passage of salt and electrolytes between the cell and outside the cell. This is followed by the plasmids, and the plasmids here, this is a very important part of a DNA or fragment of DNA within the bacteria, and it is involved or it's responsible for bacterial resistance. It was found or was thought that the plasmid is involved in antibiotic resistance, and this is the importance of plasmid, that it is a part that is not present in other cells except the bacteria. Finally, there is the flagella. These flagella are this long tail are called the flagella, which allow the bacteria to be to move, and this is one of the virulent factors that will give the bacteria a momentum for pathogenicity. It will allow the bacteria to move from one side to other, so that it can invade distant tissues. Then finally, there is some projections coming out from the cell wall. These are called the pili. These pili are important for conjugation or reproduction of the bacteria, so that the bacteria can reproduce and increase in number. So structural cell wall, cell membrane, then the plasmid, which is a fragment of DNA for antibiotic resistance, flagella, and the pili. The morphology of the bacteria can take many forms. It can be either in the form of globular shape forming cocci, or it can be rod-like like bacilli, or can be thread-like like spirochetes, or it can be a rod that has a tail. It's called the vibrios or spirella. Different morphologies will give rise to different kinds of bacteria that will help it to identify the bacteria in stains or in cultures. Regarding the metabolism of the bacteria, they can be classified according to their metabolic activity or their metabolic activity in order to produce energy into two forms. The first one can produce energy by respiration, and the other form can produce energy by fermentation. The bacteria that has been able to work to produce energy through respiration or use oxygen to produce energy are called the aerobic bacteria. So the aerobic bacteria can work by respiration, while the bacteria that work by fermentation are called the anaerobes. So the anaerobes doesn't require oxygen in order to process their metabolic actions. But there are other bacteria, or there are bacteria that will combine both mechanisms, so they can do respiration or fermentation, and this bacteria are called the facultative anaerobes. Please note that all bacteria need carbon dioxide to initiate growth. So, however, they are aerobic or anaerobic or facultative, they need carbon dioxide. But the question is, how do these bacteria produce or get their carbon dioxide in order to grow? So here the bacteria will be different, meaning that there are some bacteria that are called the autotroph. Autotroph bacteria can just consume simple inorganic compounds to produce organic substance as glucose. So, for example, they can use the light. They can use very simple inorganic compounds to produce their food. So they can cook their food. This is what is meant by the autotroph. They can made their own food by themselves, using just a very simple inorganic compounds to produce their carbon dioxide. In contrast to the autotroph are the heterotrophs. And the heterotroph, no, they cannot produce their own food, so they can use other living organisms to produce their organic substance. So in order to produce the carbon dioxide, they need to live on a living tissue. They need to live in a human being, need to live in a bacteria, need to live in a plant, in an animal. In order to use this living organism resources to produce their carbon dioxide and in order to grow. So this is what's called the difference between the autotroph and the heterotroph. And also there are some bacteria that can show the both characters. So these like divergent bacteria that can be either autotroph or heterotroph. They can sometimes they can depend on themselves for production of carbon dioxide, and sometimes they need a living organism in order to depend on it for production of their carbon dioxide. Now, after we talk about the bacterial structure and the basic characters, let's talk about the bacterial virulence or pathogenicity. How can the bacteria become invasive? What are the degree of pathogenicity of this organism? Pathogenicity of the organism. So how can this organism be harmful to the body? So the more virulent, the more harmful of the organism. There are different factors that will enable the bacteria to be virulent. First of all, the spore formation. Some bacteria, like the Clostridia, has the ability to form spores. These spores become embedded in the soil, can live for a long time without the need of oxygen, without the need of any source of energy, and they can resist the heat, can resist the trauma, can resist the environmental factors. So if the bacteria has the ability to make a spore, then this spore will be resistant and will enable the bacteria to re-invade the human tissue or re-invade the living organism and become pathologic. This is in contrast, for example, to the viruses, because the viruses cannot live outside a human body, or they cannot live outside a living organism. Also, the mutation will be another important virulent factor, so the bacteria can sometimes change their genetic information in order not to be recognized by the immune system. Infection and the tissue invasion. Also, if the bacteria has the ability to infect and invade the tissues, it's becoming virulent, and this can also be maintained by enzyme formation. Some bacteria has the ability to produce enzymes like catalase, like coagulase. These enzymes will facilitate the penetration of the organism, and it will be making it more invasive. Another important virulent factor is the toxin. Some bacteria have the ability to produce toxins, and these toxins can be either exotoxin, which is more characteristic for the gram-positive organisms, or endotoxins, which are more characteristic for the gram-negative organisms. So what are the difference between them? The exotoxins are toxins produced by the cell and released outside it at a distance. So these organisms will be produced outside the cell and will be a part of their growth and metabolism. So this is just some enzymes inside the cell that can be produced by the cell and released outside it. Like for example, the as I told you, the coagulase and the catalase enzymes are called exotoxins. However, the endotoxins, which are part of the gram-negative bacteria or characteristic for gram-negative bacteria, the endotoxins, as it is called endo, it is an integral part of the cell wall. As I told you, the lipopolysaccharide cell wall, when it is toxigenic in nature. So when the bacteria dies, the cell wall or the cell of bacteria will break apart, leading to release of this endotoxin, which can cause problems, even up to shock for the patient. So the endotoxins are released at their site or their location, destruction or death of the bacteria. So they will produce their destroying effect on the site or on the location where the bacteria are present. So this is what's called the difference between the exotoxin and the endotoxin. Again, the exotoxin is released by the cell, while the endotoxin, it is so the cell will be living in order to produce exotoxin, while in endotoxin, the bacteria should be die or should be dead in order to release the endotoxins. Now we have come to how to identify the bacteria. We have different stains and cultures used to identify the bacteria. Let's start first by the most common and most commonly used stain. It's called the gram stain. In gram stain, this stain can violet color or red color. So it stains the gram positive with the violet color or the blue color, while the gram negative are stained by the red color. So what is the process? Sometimes in exam they ask you about the process of gram staining. First of all, or the first stain to be put is staining with the crystal violet. So the crystal violet will stain both the gram positive and the gram negative, then staining with iodine, then decolorizing with acetone, then counter staining with the carbol, fuchsine, and let's see what is the process in this video.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of grams iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the...
So is it clear now what is the role of each dye? So the crystal violet will stain both the gram-positive and the gram-negative with violet color. Then, as we saw, the role of the iodine, so the iodine will remain or will maintain the stain of the crystal violet for both the gram-positive and the gram-negative. But as you said, it will make the gram-positive retain the dye and will not lose it. So when we decolorize with acetone, only the gram-negative will decolorize. So the acetone will be the differential step. This what will make the gram-positive and the gram-negative differentiate from each other. Then in order to stain the gram-negative by the red color, we counter-stain by another stain called oxime. So this will result in that there are different bacteria with different forms, like they can be present in different shapes, but the positive will be more bluish in color, while the negative will be more reddish in color. So by knowing the color of the bacteria and by knowing the shape of the bacteria, if it is cocci or bacilli, or they are threads or vibrios, we can know if they are what is the nature of the organism. Gram stains is not the only stain, but there are other stains because there are some bacteria that doesn't show or doesn't stain well with the gram. So what are these stains? Or as we said, the gram-positive and the gram-negative, they can be stained with the gram stain, and they can be also, or the gram-positive and the gram-negative can be cultured on the blood agar and the chocolate agar. Just hold on. So this is the stains, and this is the culture media. So gram-positive and gram-negative again can be stained by the gram stain, while they can be cultured by the blood agar and chocolate agar. The anaerobes, there are no specific stains, but they can be cultured and identified by a specific medium called the meat broth. The bacteria that's called nocardia can be stained by the modified Ziehl-Neelsen stain, while the mycobacterium, like the tuberculosis, can be stained by the full Ziehl-Neelsen stain. The mycobacteria, this is can be cultured on Loewenstein-Jensen medium. This is a culture medium. The fungi can be stained by the acridine orange or periodic acid chaff or the immunofluorescence, and they can be cultured on the Sabaro agar. The acanthamoeba can be stained by the periodic acid chaff and the immunofluorescence, and can be cultured by non-nutrient agar seeded with E. coli. So I just wanted to summarize different important organisms, including bacteria and fungi and the protozoa like the acanthamoeba, and their important stains and important cultures, because this is one of the common questions in the exam. Now let's move to the commensal ocular flora. So the eye itself was sterile in utero, and acquire normal flora during birth. So as it passes through the birth canal, it will acquire some normal flora that is not pathogenic. These organisms are important to identify. The commonest or the most frequent organism is the Staphylococcus epidermidis, which is found in about up to 50% of eyes. Staph aureus present in 25% of eyes. The diphtheroids are present between 25 to 40% of eyes. Staphylococcus viridans is present in a minority of cases in 2 to 3% of eyes, as well as the Streptococcus pneumoniae in 2 to 3% of eyes. And finally, the Propionibacterium is the most common anaerobe present in the eye. Again, the Propionibacterium is the most common anaerobe of the eye, and sometimes these organisms can be pathologic or can be antigenic or can be virulent, leading to ocular infections. So again, what are the commensal ocular flora? You may be asked, giving you some examples about the ocular flora and tell you which one of the following is the ocular flora except. So you need to know what are the commensal ocular flora. It is Staphylococcus epidermidis, Staphylococcus aureus, the diphtheroids, the Staph viridans or Streptoviridans, the Streptopneumoniae, and finally the Propionibacterium. After we saw the common features of the bacteria, let's talk about the classifications and what are the common ocular pathogens. So the commonest classification of the bacteria is to classify them according to the gram stain. So according to the gram stain, they can be either gram positive or gram negative. The gram positive and the gram negative, each one of them can be further subclassified into, according to the shape, into cocci and are the most important are the staphylococci and the streptococci. So they are cocci in shape and the bluish in color by gram stain. While the bacilli are the Bacillus species, the Clostridia, the Listeria monocytogenes, and the Corynebacterium. So these appears as bacilli or rods, but they appear in bluish color. The gram-negative bacteria will be cocci. It is only the diplococci of Neisseria that is present as cocci, and the gram-negative appears in the red color. While the bacilli are many, there are the group of enterobacteria like the E. coli, the Klebsiella, and other organisms. There is the Pseudomonas and the Legionella and others. And there is a group also that is not taking the gram stain, that are the mycobacteria and the anaerobes. So they have special stains and cultures. Let's first talk about the gram-positive bacteria. What are the gram-positive bacteria? We said we have cocci and the bacilli, and every one of them is further subclassified if it is aerobic or anaerobic. It can live with oxygen or it can live without oxygen. So the aerobic bacteria can be either present in chains and during a culture, which is the streptococci, or present in clusters in culture as well, as in case of staphylococci. So the aerobic cocci are staphylococci and streptococci. There is no anaerobic cocci, and in bacilli there is aerobic bacilli. Can be either spore forming or has the ability to produce spores like the Bacillus species, or they don't possess spores or they can process spores like the Propionibacterium. The anaerobic bacilli can again be further subdivided into spore forming like the Clostridium or non-spore forming like the Actinomyces. So this is the classification of the gram-positive bacteria. Let's now talk about each one of them and know what are the main features and how does each bacteria of them can produce the ocular pathogenicity. Let's talk about the staphylococci. Staphylococci, as we mentioned, it is a gram-positive aerobic cocci that is present, like here. There are some characteristic features or some virulent factors regarding the staphylococci. So the staphylococci are catalase positive. All staphylococci has the property of catalase enzyme, or has the ability to produce the catalase enzyme. This will be in contrast to the streptococci. The Staphylococcus aureus also have the ability to produce the coagulase, making it very virulent. So coagulase positive is present in the Staph aureus. On the other hand, the Staphylococcus epidermidis is coagulase negative, which is one, as we said, it is the commonest ocular flora. So remember that since the staph epidermis can be one of the ocular flora, it doesn't have this virulent factor of coagulase enzyme, so it is coagulase negative. While the staph aureus, it is a dangerous or a severe infection, can be producing keratitis and other ocular infections. It is a coagulase positive. There is a specific type of staph aureus called the methicillin-resistant staph aureus. The methicillin-resistant staph aureus is, if you remember it from the pharmacology lecture, we said about it that it is can be treated by the cloxacillin. It is found in the anterior nares of the asymptomatic carriers, although it is a severe infection, but sometimes it is found in the nares or the nose nostrils of a patient or of a normal individual who is asymptomatic. So these are the important features of the staphylococci. And as we said, it is by the gram stain, or to identify it just by staining, you will find there is a gram-positive cocci, as you can see here. And by culture, they can be cultured on they will produce like golden yellow colonies. The second of the list is called streptococci. Streptococci again is a gram-positive cocci that is catalase negative. So remember which one is the catalase positive and which one is the catalase negative, because it is asked frequently in the exam. They are uncommon ocular pathogens, so it is rare to find a patient who or an ophthalmic patient who have a streptococcal infection. So we don't see it on a regular basis. The Streptococcus pyogenes is responsible for 90% of the Streptococcus infection, so this is the commonest cause of streptococcal infection. The most common organisms to produce, or the commonest diseases produced by the streptococci are produced by the beta hemolytic streptococci, which produce an erythrogenic toxin called leading to erythema of the skin, caused what is called by the scarlet fever. And there is another form of the streptococci that's called alpha hemolytic streptococcus pneumoniae, or a streptococcus pneumoniae that is alpha hemolytic and will produce meningitis and pneumonia. So these are the different species or the different types of streptococci that will cause the different diseases. Remember that the pyogenes or the streptococcus pyogenes is the most important and the most common infection. And the beta hemolytic streptococci will produce scarlet fever, while the alpha hemolytic streptococci, or what's called streptococcus pneumoniae, will produce meningitis and pneumonia. Remember, it's catalase negative, and remember that it is also gram-positive cocci present in the stain and can be cultured on the blood agar. Then, after talking about the gram-positive cocci, which are aerobic, let's talk about the gram-positive bacilli. So the gram-positive bacilli start with the Bacillus species. So Bacillus species are gram-positive bacilli, appear bluish on gram stain. They are aerobic, and they have the ability to form spores. So these spores are one of the virulent factors, as we mentioned, that will enable the bacteria to reproduce and stay alive outside the living organism. until they have a chance to invade this living organism again. Different species of the bacillus are called the Bacillus anthracis, which is known for producing the anthrax, this respiratory infection, in which the Bacillus anthracis will have the ability to produce the exotoxin. This exotoxin will produce the anthrax. The Bacillus cereus, which again has the ability to produce an exotoxin that will cause a food poisoning. And finally, the Bacillus brevis, from which we extract the antibiotic, the bacitracin. So these are the main features of the bacillus species. Again, they are aerobic, they are spore forming, produce exotoxin. The Bacillus anthracis produce exotoxin that will cause anthrax. Bacillus cereus will produce a toxin that will produce food poisoning, and the bacitracin will be extracted from the Bacillus brevis species. Another form of the gram-positive bacilli, it is called the Clostridium. So Clostridium are anaerobic and spore-forming. So in contrast to the Bacillus species, they are anaerobic, while the Bacillus are aerobic. They have the ability to produce spores, so there are some different species in the Clostridium. We have the Clostridium tetani, which produce exotoxin that cause tetanus. This is very important. So the tetanus is a neurotoxin that will pass from the Clostridium. So basically, what happens is if someone is not taking the tetanus toxin or the tetanus vaccine, then this patient is susceptible to trauma. For example, if he injured his foot by nail or by whatever in the ground, and this object or the soil is contaminated with the spores of the Clostridium tetani, then the spores will invade the human tissue and produce their powerful exotoxin, which will pass into the CNS, causing paralysis and causing the tetanus, leading to even death. So it is a fatal disease unless the patient has the toxide or has the antidote. So this is called the Clostridium. Another serious form of the Clostridium is called the Bacillus perfringens. Bacillus perfringens is again a gram-positive bacilli that has the ability to produce spores. When invade the tissue, it can produce exotoxin that is responsible for gas gangrene. And this is a very serious and fatal disease that will lead to death. And this infection, if it is present in a ward or in a hospital, usually this hospital is closed because this is a very serious infection. There is some strains of what concerns us as an ophthalmologist, that there are some strains of Clostridium perfringens will lead to conjunctivitis, necrotizing keratitis, and suppurative panophthalmitis with retinal necrosis. So as you can see, this disease is very, very catastrophic and disastrous, leading to severe infection. You see that there is a lag between the voice and screen. All of you have this lag? I don't know what to do about this, as I have nothing to do, but hopefully that it will be improved in the recording. So I guess that we have no option but to continue. Okay, so another form of the gram-positive bacilli is called the Corynebacterium. What are the Corynebacterium? They are gram-positive bacilli. Again, they are aerobic, they are spore-forming, and they live as commensals on the skin and the mucous membrane. So these are called the Corynebacterium. Gram-positive bacilli live as a commensal on the skin and the mucous membrane. They are aerobic and spore-forming. So I don't want to overwhelm you with a lot of unnecessary information, because this is so I just want to stress on the important points about every microorganism. Another important and very important organism that has an ophthalmic correlation is called the propionibacteria. The propionibacteria are the gram-positive bacilli that are anaerobic and non-spore forming. So we said that the propionibacterium are the most common commensals of the most common anaerobic commensal in the eye. So the propionibacterium acne live as commensal on the skin and the mucous membrane and inside the eye, and sometimes they can become a commensal normally in the eyelid and can be isolated from the eyelid and within the meibomian glands. However, sometimes they become pathogenic. So as long as they are present within the eyelid and the meibomian gland, they can cause chronic blepharitis. So this is one of the diseases produced by the propionibacterium acne, and the other most important is called the chronic endophthalmitis. Chronic endophthalmitis, it is a disease produced by the propionibacterium acne, which is not present acutely as the acute postoperative endophthalmitis, but can present few months after the initial surgery, present with white plaques in the capsule due to sequestrated organism, and present with corneal edema and granulomatous keratotic precipitates, as it can cause granulomatous inflammation. So this disease is called the chronic endophthalmitis, and the commonest cause of it is the propionibacterium acne. Finally, we have the actinomyces. The actinomyces are gram-positive bacilli that are non-spore forming. They can be sometimes considered as fungi, and we will talk about it again in the fungi. So because these, although it is a gram-positive bacilli, but they can form a mycelia or filaments. These mycelia, or if we examine it under the microscope, they will be forming a branching filaments like this, which appear as a gram-positive bacilli. So they can be similar to gram-positive bacilli or similar to the filamentous fungi, and the commonest species of it are called the Actinomyces israeli. The Actinomyces israeli are anaerobic organisms causing a canaliculitis, leading to what's called the pouting punctum, and also can cause dacryocystitis. So Actinomyces israeli is producing an anaerobic infection causing a canaliculitis and dacryocystitis. The other species of the Actinomyces is called the nocardia asteroids, and this has been asked before in the exams. The nocardia asteroids has the ability to produce a severe keratitis and endophthalmitis. So the nocardia is causing keratitis and endophthalmitis. This is all what you need to know about the actinomyces. So this concludes the gram-positive bacteria. Let's now talk about the gram-negative bacteria. We have different types of gram-negative bacteria according to the shapes. As we said, we have the gram-negative cocci and the gram-negative bacilli. Each one of them is further subdivided into aerobic bacteria and anaerobic bacteria. And finally, we have another group of bacteria that can be considered gram-negative but doesn't belong to the cocci or bacilli. They are called the spirochetes. So let's start by the cocci. What are the aerobic bacteria? There we have the Neisseria. So the Neisseria can be considered a gram-negative aerobic diplococci. There is no anaerobic cocci, and the bacilli, we have the aerobic bacilli are the Haemophilus species and the Pseudomonas species. And finally, the anaerobic are the Campylobacter. The Campylobacter bacteria are considered anaerobic bacilli, gram-negative bacilli. And finally, the spirochetes like the Treponema, responsible for syphilis, the Leptospira, and Borrelia. These are called the spirochetes. And they have a characteristic shape and pattern in examination, as I will tell you later. So let's start first with the Neisseria. The Neisseria are gram-negative diplococci, so they are present in pairs. Two pairs of red balls are called the Neisseria. The Neisseria are gram-negative diplococci that are aerobic, that consist mainly of two species. The first one is called the Neisseria meningitidis or the meningococci, and the other one it is called the Neisseria gonorrhea or the gonococci. Since we are now talking about the gram-negative bacteria, this bacteria will possess a endotoxin activity. So as we mentioned, these bacteria can be virulent by their endotoxin activity. Also, the Neisseria has the ability to produce a virulent enzyme called the IgA protease. So what are the pathological effects of Neisseria? What concerns us more in ophthalmology is the Neisseria gonorrhea, or the gonococci, because the gonococci, when they are transmitted from the mother to the patient or to the child during birth, can cause the characteristic feature of ophthalmia neonatorum. The child will present with purulent conjunctivitis, with purulent discharge, and this disease is a serious disease because it can lead to corneal perforation. So it should be identified and be managed appropriately. In order, if we are in doubt of diagnosis of Neisseria, even with the staining on the gram stain, we can culture on a medium called the Thayer-Martin medium. So Thayer-Martin are used for the second on the list, or a gram-negative aerobic bacilli, are called the Pseudomonas. The Pseudomonas are gram-positive aerobic bacilli. This disease is very serious and very virulent organism that not only can infect the eye, but different tissues of the body. However, although it is virulent, but it is non-motile. The importance about the Pseudomonas is that it can progress very rapidly. So it can invade the cornea, but luckily enough, it doesn't invade the intact epithelium or doesn't penetrate the intact epithelium. In order for the Pseudomonas to penetrate the cornea, the epithelium should be traumatized. So once the epithelium is traumatized or once the epithelium is injured, then the organism can produce their protease enzyme, and the protease enzyme will cause the severe keratitis. Sometimes this keratitis can progress into 2 days in as soon as 48 hours into corneal melting. So this disease is very serious, and I have even saw some patient with Pseudomonas keratitis that progressed to endophthalmitis and the panophthalmitis and ended by not only inoculation but by exenteration because of the Pseudomonas infection. It also has as I told you, it will produce an enzymatic activity by the protease enzyme, and also produce a toxin that's called toxin A. Another gram-positive bacilli is called the Haemophilus influenza or Haemophilus species are gram-negative bacilli that are aerobic, like the Pseudomonas, so they fall on the same category. And there are different species like the Haemophilus influenza that will be important in producing orbital cellulitis. It is very common in children to have sinusitis, and this sinusitis can spread to the orbit, leading from the sinus, from the maxillary sinus, or more commonly from the ethmoidal sinus, to the infection to reach the globe or to reach the orbit, causing orbital cellulitis. One of the commonest organisms to produce this picture are the Haemophilus influenza. There is another organism called the Haemophilus aegypticus. So Haemophilus aegypticus has been associated with conjunctivitis. And finally, the Haemophilus ducreyi, which is present with a chancroid, which is a picture similar to the chancre of the syphilis, and the perinode oculoglandular syndrome, which is present with follicular conjunctivitis and preauricular lymphadenopathy. So you need to know what each organism, what is the disease associated with this organism. So again, Haemophilus influenza with the orbital cellulitis, Haemophilus aegypticus with conjunctivitis, and Haemophilus ducreyi with the perinode oculoglandular syndrome and chancroid. Moraxella is a gram-negative diplobacilli. It is aerobic and can produce the picture of infection of the angle of the lid and the conjunctiva, causing what's called angular blepharoconjunctivitis. Angular blepharoconjunctivitis, it is a disease characterized or caused by the Moraxella species. And also it has the ability to produce corneal ulceration and keratitis. Enterobacteria are gram-negative bacilli that can be either aerobic or can be facultative anaerobic. So they can be either aerobic or anaerobic. There are present as normal commensals of the GIT like the E. coli. The E. coli itself is the commonest cause of urinary tract infection. And in order to culture it, this can be done by on agar called the McConkey agar. So the McConkey agar used for culture of the E. coli. And there is an important species of the enterobacteria that also was asked before in the exam. It's called the Serratia maricensis. Serratia maricensis are, it is a drug with corneal inflammation because it can contaminate the contact lens solution, leading to keratitis and endophthalmitis. And the problem with it, that it is difficult to diagnose. So the patient will pass for maybe misdiagnosed as acanthamoeba keratitis for a long time, as he is a contact lens wearer, and so the proper management for the Serratia maricensis will be overlooked. Now we talk about the spirochetes. So the spirochetes are a specific category of the gram-negative bacteria that have a specific pattern in the examination, that have a helical structure. You see that this organism is coiled upon itself. This has a helical structure, or they have a long thread, very long thread, and this helical structure or this flagellated structure will give the organism the ability to move, so it will allow it for spiral motility. So again, the spirochetes are organisms that have helical structure that will be this helical structure will enable it for spiral motility. They can be visualized by the dark ground microscopy, and one of the most important features here are called the Borrelia burgdorferi. Borrelia burgdorferi, it is an infection or a spirochete that is transmitted by bite from the deer tick in the tropical areas. That will cause a disease called Lyme disease. In Lyme disease, there is the first stage and second stage and third stage. The first stage is characterized by what's called the target lesion. The target lesion is at the area of injection, at the area of bite of this deer tick, and this is followed by stage 2 and stage 3, in which the patient will report ocular complications, specifically or most importantly uveitis. Then another form of the spirochetes are called the treponemes. So the treponema, as you can see here, this is treponema, again has a helical structure and the flagellated structure that will allow it for spiral motility, and again it can be visualized by the ground microscopy. So the most important of the treponema are the treponema pallidum, and the treponema pallidum is the causative organism of syphilis. And in order to diagnose the syphilis, the gold standard for diagnosis is serology. So there was a previous previous serological tests used for diagnosis, like the Venereal Disease Research Laboratory, or with the VDRL, but this disease is now not considered the gold standard because it lacks some specificity and sensitivity. And the gold standard now is the fluorescent treponemal antibody absorption, or FTA antibodies, or FTA absorption. This can be considered the most sensitive test for the diagnosis of treponema pallidum infection. Then another specific category of gram-negative bacilli are called the gram or the acid-fast bacilli. The acid-fast bacilli, like the mycobacteria, are called acid-fast and alcohol-fast. These organisms are aerobic. They doesn't produce any spores, and they are rods, as you can see here. So they appear in reddish color, and they are aerobic, non-spore forming, and they are stain, or the specific stain for the acid-fast bacilli or the mycobacteria, it's called the Ziehl-Neelsen stain. I think that we remember this from the undergraduate study. So the Ziehl-Neelsen stain is the specific stain for the mycobacteria, and the specific culture will be on the Lowenstein-Johnson medium. Lowenstein-Johnson medium is the specific culture medium for the acid-fast bacilli. There are different species of the acid-fast bacilli that produce different diseases, like the Mycobacterium tuberculosis that will produce the caseating granuloma of the tuberculosis. There is Mycobacterium leprae, which will produce leprosy. Mycobacterium avium and the Mycobacterium fortitium, which is producing a corneal ulceration and endophthalmitis. The Mycobacterium tuberculosis will be associated with tuberculosis. Mycobacterium avium will be Mycobacterium leprosy will be associated with leprosy. Mycobacterium avium and Mycobacterium fortitium will be associated with the corneal inflammation and ulceration, endophthalmitis in the immunocompromised patients. So is everything clear up till now? So I just forgot to tell you that the acid fast and alcohol fast meaning that they resist the decolorization by the acid. So acid fast means that it resists the decolorization in the lab by the acid, and the alcohol fast also they resist the decolorization by the alcohol. Just as a quick note about the acid fast bacilli. Okay, so let's now move to the viruses.